Hi everyone, welcome back to class. Today we're going to go over kind of like the second set of background info that you need to know before we start on specific drugs in this course, and that is pharmacodynamics. So remember in the last lecture we talked about what is pharmacology and pharmacokinetics. We will now be talking about pharmacodynamics. We'll also do a really quick review of drug tolerance, and I do mean very quick. A lot of this you should have, if you've taken a pharmacology course, a lot of this should be familiar, and some of these topics you may have encountered before in other psychology classes as well. So a lot of this should be review. So what exactly are pharmacodynamics? This is the study of the physiological and biochemical interactions of a drug molecule with their target tissue and the receptors responsible for their ultimate drug effects. So this is really focusing on those specific drug effects. We're kind of ignoring the non-specific drug effects now. Now, this does contrast with pharmacokinetics a bit, which is the factors and processes that affects the bioavailability of the drugs. So pharmacokinetics is the availability of the drugs, whereas pharmacodynamics is the actual drug actions. So this is how the drugs interact in the body. This is what we're going to be talking about for most of the class. So we have different drug receptor interactions that occur in the mammalian body. First, you should know what a ligand is, and you should also know what a receptor is. As a small review, a ligand is any molecule that binds to a receptor. So it can be a drug, it can be a hormone, it can be a neurotransmitter, it can be a protein. It doesn't matter. It's anything that binds to a receptor. There's two types, of course. We've got exogenous ligands, so like a drug, something that comes from outside the body, and then endogenous ligands, so anything from inside the body. Okay, examples, but inside versus outside. A receptor is a protein molecule. It's usually made up of multiple proteins. It's on the surface of a cell or sometimes within the cell itself, and that is what obviously ligands bind to. It is the initial site of all drug actions. Okay. It is the site of action for endogenous and exogenous ligands. Sometimes I might just call, say drug, and I'm talking about a ligand. Okay, so drugs, what we think of as drugs are, more things that we take. But you can also think of neurotransmitters or hormones as your body's natural drugs. Okay, so we largely discuss drugs as ligands for receptor molecules. So like the case with psychoactive drugs. Effectively, any cellular protein is capable of acting as a binding site for these drugs. So a useful example using psychoactive drugs would be antidepressants. So MAO inhibitors bind to monoamine oxidase, which is a metabolic enzyme, whereas SSRI antidepressants bind and inhibit the serotonin transporter, which is a membrane transporter. So there's two different types of antidepressants. So it's a ligand that does the same thing, but it targets different receptors. So some receptors are inside the cell, so like a metabolic enzyme is going to be inside a cell, whereas the serotonin transporter is a membrane transporter, so that's going to be on the cell surface. Okay, so two different ligands can create the same or have the same drug effect, even if they act on receptors in different parts of the body or different parts of the cell even. Okay. So, how do we define a receptor then? Well, since there's many different types of proteins and enzymes that are capable of binding ligands, then it's very useful to define the characteristics of our receptors. So we do that using kind of these three big things. Number one is saturability. So this is a finite receptors per cell. So it's really just the number of receptors per cell. Dose response should reveal saturability. In other words, there should be an amount that at some point you can't, it doesn't matter how much drug you give, there's a ceiling to the effects of the drug. And that's just because there's no more receptors available. Okay, there's only so many, it's finite. Now, saturability can change over time, depending on LTP and LDP, but we tend to think of saturability as finite. The number two thing that can define a receptor is its specificity. So a high binding affinity to elicit a biological response. So some receptors, it only takes a very specific drug or ligand to bind, and then there's other receptors that have less specificity and will take really any ligand that meets a broad criteria. An example of that would be MAOIs. We'll talk about them, but it's a monoamine oxidase inhibitors. And so those can go and bind to, any monoamine, and there's 3-4 monoamines. So that specificity is different than, say, just a serotonin reuptake inhibitor. Okay, those are going to be different. And then reversibility. So binding to a receptor should always be reversible. So in your natural drugs or ligands that your body produces, the ligands will bind to the receptor and then unbind. They will leave. They will be dissociable and recoverable. So we can then recycle, reuse those ligands. If something binds to something else, to a protein, and then it sticks there forever and ever and it can't get off, that is different, okay? So this distinguishes the receptor-ligand interactions from an enzyme-substrate interaction. So an enzyme-substrate interaction, then what the enzyme does is it, binds to the substrate, and then they're just bound. That's it. They don't unbind. So here again, we've just got receptors, drug or neurotransmitter, so some kind of ligand. Boom, it binds the receptor. There's an effector on the inside of the cell that creates those intercellular effects when a ligand binds. Then here we've got a receptor that's inside the cell. So here maybe a steroid hormone is the ligand. It's going to have to pass through that cell membrane and then bind. And then the hormone receptor complex that it creates will then travel and then have an action someplace else. So these are the two distinct classes of receptor binds that are either water-soluble or lipid-soluble ligands. So these extracellular receptors are on the cell surface, and this is really for those water-soluble ligands. So those more lipophobic ligands, and then lipophilic receptors can cross that membrane easily, so their receptor can be within the cell. So that kind of determines where your receptor is going to be, is the solubility of your ligands. We, of course, have those receptor types, and there's more information about them. So those extracellular receptors, very common for psychoactive drugs. There are different signalings based on the function. So we've got ligand-gated ion channels. These are very simple. Ligand binds, channel opens. Boom, bang, done. Great. These are almost always on that postsynaptic neuron. We then have G-coupled protein receptors, and these are very common, especially when we start talking about GABA and glutamate. Okay. And then we have receptor kinases. So these are kind of the, in terms of popularity, ligand's the most popular. G-coupled protein is going to be your second most popular, and the third one's going to be your receptor kinases. So these are common for cytokines and peptide hormone receptors, but we're not going to talk about very many of those in this class, but they will come up again once or twice. So you should still know that that is a third type, and that they are located on that cell surface. Then we've got our intracellular receptors, and these are very common targets for hormones and lipophilic compounds. Now, some drugs do go there, but most drugs tend to bind extracellularly. Some examples of those hormones would be glucocorticoids, so the stress hormones, sex hormones, and then there are actually some intracellular G-coupled protein receptors. So those would be endocannabinoids. So we will be talking about endocannabinoids when we get to the cannabis section near the end of the semester. These receptors really just float around in the cytoplasm, just hanging out, waiting for some binding. Hormone receptors are, they're the specific type of receptor we'll talk about as these intracellular receptors. They translocate to the nucleus. In other words, they move. Once they get their passenger, pretty much, of the hormone, then they will move into the nucleus itself, and then it will function as a transcription factor. So it causes transcription to occur. This can then directly influence the changes in the gene expression of the cell by binding to specific response elements. So different hormone receptors will bind at different places in the genes to create specific response elements. These elements are located in that genomic DNA specifically, and it's actually a pretty cool system. Now, let's just go over, the receptor-ligand interaction. It should be pretty simple. It's pretty a lock and key thing, but there's different things that can occur at this receptor-ligand interaction site. And that's when we start talking about agonists and antagonists. So these are ligands that will bind to the receptor, but aren't a natural ligand. Okay, so an agonist kind of eggs it on. Okay, it turns it on. So that's what this blue example is up here. It's pretty much like it's a key that it's a copied key. It's not the key that your landlord gave you to your apartment. It's one you went and made. So it's like a fake key, but it works. So it still will bind to the receptor on the cell surface, and it will cause that receptor to have a drug action. A drug effect will occur. Okay, so these elicit a biological effect. The other type would be an antagonist. So an antagonist is still going to be a ligand that binds to the receptor. However, what it's going to do, it doesn't unlock the key. It's pretty much like if you force the wrong key into a lock, and then that wrong key kind of gets stuck there. Okay, so it doesn't unlock the door, but you can't get the key back out. That's pretty much what an antagonist is. It's going to bind to the receptor to prevent drug action. Okay, so it's going to block a agonist or the proper ligand from binding. So, while not all drugs target receptors, some do target like tissue and stuff, the general principles of specific ligand interactions is the same. So there is always a lock and a key of some sort. So here we are really just assuming that a receptor can be generalized for any drug target. So just keep that in mind. So why do we use agonists? Well, one, sometimes, we just don't have enough of the natural ligand that our body creates, so we want to increase it. So we have to create a copy. And sometimes, recreational drugs, a lot of those will be agonists. So agonist activity at a receptor can actually be measured by examining the dose response. So that's what this graph here is it's a dose response chart. On the x-axis here, we have the dose. So as the dose increases, we go to the right. And on the y-axis, we have the percent response. Okay, so from 0 here to 100. With increasing the concentration or increasing the dose of the agonist, the biological response increases. Awesome. But you'll also notice that there seems to be a maximum response. There's some point in which we can give them more drugs, but there's just nothing left to give, right? And that goes back to that the receptor saturability, right? They're fully saturated. Giving more drug isn't going to do anything. Now, you'll notice that this is kind of like an S shape. Okay, if we start at the top, it's kind of an S. This is what we call an S curve or a sigmoidal shape. It's just very distinct. Right here, then this is about 50% response. Okay, 50% response. That dose, you'll see here, is labeled ED50. Okay, so ED50 is a convenient measure used to compare the potency of a drug. So this is the dose required to elicit half of the maximum effect. Makes sense. It's at 50% response. Okay, ED50. It's very an important thing. I know you'll see why. But note that ED100 is not useful. Okay, we don't use ED100. Why? Because usually by the time you hit ED100, you're dead. Think about some illicit drug. Okay, so like heroin, cocaine. If you can get to ED50, you're probably not alive. Now, of course, there are some drugs, that you could get to ED100, but it doesn't tell us anything. Okay, it's very impractical. And that's why we use ED50. And this is what we use to determine potency. Now, there's some other kind of things you should know about. But what we can do is compare the potency of different drugs by comparing their ED50s. Okay, so here we're looking at different opioids. Okay, so we're comparing the curve shape and a dose response plot. So ED50 shows us potency. However, all three opioids have the same efficiency. Okay, so efficiency is the maximum effect possible when given a drug. So you'll see here that the ED100, if we were to get there, gets to 100 up here. So this is the percentage of elevation in pain threshold. Okay, so there's 100. It can get to 100. All three of these, hydromorphine, morphine, and codeine, can all get to 100. But their ED50s, you'll see, are different. This one has an ED50 of, say, that's probably a 2. Morphine has an ED50 of 10. Codeine has an ED50 of 100 and something, right? So they have the same they have different potency because they have different ED50s, but they have the same efficiency because they have the probability of getting to the top, of getting to 100. Now, if we look at aspirin here, that's the one in red, it has both a different potency and a different efficiency. Okay. First, efficiency. That differs from all three of these. Why? It can only get to 75%. You'll see that's the maximum on the X, or sorry, on the Y. Okay. It cannot get to 100, so it has a different efficiency. Probably, we'd say a lower efficiency. It would also have a different ED50. ED50 would probably be right about, well, it's right there. Our lines are helpful, so it'd be up here. What is that dose? I don't even know, because this is not to scale. But it doesn't overlap with any of these lines, so we know that it's going to have a different potency. Okay, so the difference in efficiency and the slope and width of this curve, notice it looks different, suggests that aspirin probably works through a different mechanism, whereas hydromorphine, morphine, and codeine all probably work the same. Okay. Now, let's stop there, get your head around potency and efficacy, and then when we come back, we'll start with the therapeutic index, because we're just going to add even more stuff and talk about math a bit.